Hi guys. So we had our NGS workshop last week, and unfortunately, we weren't able to record due to technical difficulties. And this is something that um, will be really useful to students who are interested in next generation sequencing in bioinformatics field. And I've received multiple requests from students who were unable to attend the workshop due to classes and homework commitments. Um, so I just have decided to quickly um, go over the workshop that we did last week and give you the keynotes that you need to know for the pipeline. For those who have never been to the NGS workshop, I highly recommend that you check out our first NGS workshop, which is also uploaded on the YouTube page. So you'll get an idea of how we got the data and what the pre-processing step we have already done to get to the alignment stage. Yeah, without further ado, let's get into it. So I will quickly talk about the alignment and peak calling for this workshop. So go to the next slide. Yeah, quick recap. So we did the adaptive trimming after we downloaded the FASTQ files from the NCB SRA, and we downloaded the Daniel genome. That was our data set, and we performed the indexing of the genome last workshop. In this workshop, we will do the alignment using HiSet2, and I'll talk about the HiSet2 in a little bit. And then we will talk about the same format that we will get sequence alignment map from after the alignment. And then we will look at the output of the alignment file. So here's a quick flowchart or roadmap for today's this presentation. So FastQC, then you do the alignment, then you filter the BAM files or sort them, and then you do the max2 peak calling. So why use HiSet2? There are multiple aligners out there to do the alignment. For example, Top Hat, we have stars, we have bow tie, we have HiSet2. It's recent. So why exactly use HiSet2? Just quickly summarize, the HiSet2 is using the Burroughs-Willis transformation and FM index. And what they do is they do this hierarchical indexing by interchangeably compress the data and extract the data. And with that, because of that ability, they're able to compress their memory. And I'll talk a little bit about that in the next few slides. So a bigger idea is that HiSet2 uses small, less amount of memory. On contrary, Top Hat and star aligners, they uses this other suffix array method to keep to index the genome. And they are unfortunately not able to retain the information after converting into this array. So due to that reason, it requires more memory. Now I'll talk about that too. So quickly going over the WT transformation and FM index. So WT transformation is, suppose you have a read, some sort of sequence that you want to align to the genome. So in order to do that, you first have to transform it into WT transformation, and this is the method that you can do the transformation. And this is more compressed format because it does the alphabetical sorting, and this way it uses less space. And alongside this, it uses this FM index to retrieve the primary read, which is this read right here. Okay? So Actually, sorry, it uses this FM index to map the reads, to find this specific reads found in the given data set on the genome. So L is actually uh, our genome, and F is the reverse index. So F is the primary uh, read, and it's trying to back index. And you can watch the lecture, or actually watch a YouTube video on this, and it's better explained there if you want to get into that. But the whole idea is that this light gray area you see here is not stored, and that's the reason why we're using the less memory, right? Because it's actively is able to reverse um, engineer to get the reads that we're looking for, to map the reads that we're looking for. On contrary, the suffix array that is used by store, what it does is it indexes all these read, right? The Mississippi, for example, it indexes all these different variations of Mississippi in the binary format, and then it is used for the alignment purposes. But what the suffix array does is also retains this information, right? The read information, which is the reason why it's using or requires extensive amount of memory, which usually our local computer doesn't have, right? And that's why most of the time, if you're running this sequencing analysis on your computer, it is suggested and recommended to use HiSet2. Yeah, so that's the general idea and difference between these two. Quickly looking at the sequence output alignment map format, we have the header, reference genome information, read information, program software we're using, and the commentary lines. And they all are mentioned here. You can pause the video and look at different things. The same format specifically has multiple things. Read name, map. Map is this 0, 4, 16, 0. So 0 means okay, meaning that the read was able to perfectly align with the genome. Sorry, yeah, and the 4 means unmapped. There are regions which were unable to map to the genome, or the read was unable to map to the genome. 16 means that the read was able to map to the genome, but it was in the reverse strain. It was from the 3' end instead of the 5' end, right? Then we have sequence, quality score, etc. down here. One thing I want to mention here is this highlighted blue part. So this is showing exactly where the mutation has happened. So for example, this is showing that there's a 3 match. On the 4th index, there is a mutation. So if you look at here, right, TAC was perfectly aligned with the genome. But the 4th index was supposed to be C, which is on the genome, but the read has A. So it was not able to map to the genome. So that's why we see a mutation of C. Then next 30 base pairs were perfectly aligned with the genome. And then the 4th to the last base pair was not aligned. So 1, 2, 3, 4, 4th to last, which is G. So on the genome, it's T. So we see that there's a different base pair. It wasn't aligned to the genome. And then the last 3 were perfectly aligned to the genome. So that's the key information. Yeah. And then NM is the number of mismatches. So here we have 2 mismatches, right? We had at the 4th index here, which is the C, and the, I guess, 31st index, the T over here. So that's why we have 2 here. On this one, we have no mismatches, right? So how do we know that? 1, it says 0. 0 means okay. Second, it says all 38 base pairs of this read were mapped. And then third, it says it's 0. the number of mismatch is 0. Right? This is key information if you want to, extract information from SAM files. And there's a whole research about this they can get into. Quickly going over the binary file formats. After converting the SAM files into BAM files are just the binary index files. So once you get the SAM sequence alignment map files, you will change them to header to the binary index because they're more compressed. What you will do is what you're doing is that you're compressing all this information in different format. So this is just a quick overview of what you have here. So the header has the information file, the sequence length information, alignment information. The alignment has the read name sequence, resequences, quality, etc. And it has other things, the barcode tags, etc. for each reads. So usually when we open the binary files, we're going to see some jargons, and it is probably not going to make any sense to us. And we more often than not, we will not be opening any binary files. It is the same files that we usually edit before we start doing any further downstream analysis. So now once we have our BAM files, it's time to peak call. So before I get into the tools that is used for peak calling, what is peak call, right? Let's understand that. So suppose we have a DNA strand that we're trying to sequence. One thing that we are interested in understanding is the region of the DNA strand. Where exactly are the proteins, the transcriptional factors are binding? So for example, if the DNA is transcribing to RNA, the RNA polymerase has to come and bind to the DNA, right, in order to start the transcription. So how is that complex forming and binding to the DNA? So we're interested in those transcriptional factors, not only just the RNA polymerase, but there are other proteins which are binding to the enhancers and repressor sites of the DNA upstream and downstream to regulate the rate at which this gene is transcribed from the DNA, right? So that's why we're specifically interested in understanding this DNA protein interactions. And through these regulations, this gene can be overexpressed or underexpressed. And by extension, more proteins will be made for that specific gene or less. And their role will be impacted, right? So now that we have clear ground of what protein and DNA interactions are doing, let's get into what various tools are out there to do the peak calling. So the famous peak caller that I am using for this session is the MAX2. MAX2 is very well known for ChIP-seq, chromatin immunoprecipitant analysis, which is specifically for the protein RNA interaction of protein DNA interactions. So you don't need to know about the algorithm that it uses. Just to give you an idea of what they are doing is compared to the other tools, is they're using this dynamic Gaussian distribution to capture the local biases, which is the reason why these MAX tools are better at giving higher accuracy compared to the other tools available in the market today. It also gives you the high peak resolutions, and it uses low starting material. It can work with low starting material at lower cost. So what I mean by low starting material is, suppose you're working with only one replicate. You don't have multiple replicates, or you can't afford multiple replicates for a study. And you still want to get the output to study downstream analysis. And in order to do that, you need a peak caller which is accurately able to predict the peaks at high resolution, so that you can then downstream analyze it, yet visualize it, and make um, uh, hypothesis that can further the biology, right? So that is what Max2 is providing. Lastly, this is the output that we are expecting after we do the peak calling. So we will have chromosome information start at end position, and then we will have these three information. I don't think we'll have FRET score. We will have full enrichment information, p-value information, and FDR information. The full enrichment is just uh, log fold, I believe. The higher the fold, the better. And p-value is lower the p-value, the better. Right? And FDR is the false discovery rate. Usually 0.05 is the threshold used uh, for the study. So yeah, I will show you these quickly from the file that I have gotten after the analysis. So yeah, after we get the peak calling, what's next, right? So after peak calling, we'll do the differential binding analysis. We could do. I don't think we will be doing it for this series, NGS series, but there is an option to do differential binding analysis that shows us that um, which specific um, set of proteins put in DNA interactions are differential compared to the control group, right? And we can further look into the genomic distribution, like what regions these binding sites are located in, whether that is an exon, intron, intergenic region, or link RNA, excuse me. We can further do the gene enrichment analysis, is where we input all the genes that we have received from the max to output. So to do that, first what we have to do is download the annotation file, which has the gene information. And then we cross-reference that with this file to get all the genes associated with this region, right? So we will add a column with all the gene names from these associated coordinates. Once we have all the list of genes, then we will take those genes to build a network to understand what networks are associated with this set of genes. And then we can pick one network out of multiple different networks and study that further. And that could be a one hypothesis, right? That this specific network, for example, cell cycle, this set of 20 genes are associated with cell cycle, and they're regulating the cell cycle and controlling uh, this specific gene or this set of genes. And we can build a study on that. Another thing we will do is visualization. So we will want to understand how the treated sample is showing different expression compared to the control sample, normal sample, right? And what genes are showing different expression compared to the treated, the normal sample. And that can be done using visualization from UCSC browser and IGV. Okay, so quickly, I want to share the script with you guys. So again, this is tailored towards students at IPY. This batch, so this is S-batch is a Slurm system for our automated job submission for the supercomputer. So we are using Carbonate as a supercomputer at our campuses. So this first hashtags you see is just giving details to the job scheduler, how much, what details and computing power we are assigning it for this specific job. So quickly going over it, so you're aware of this. Every Linux file that you make, to run that file, you must include hashtag !/ bin / bash. When you're submitting a job, you need to submit the name of the job, like what name it will be registered as, and that's like I just mentioned NGS series alignment. There are different intensive computation intensive jobs, and this job is not really that intensive, so I'm giving it as general. There are other deep learning uh, jobs that you can um, um, mention if your job is more extensive. The output is what output file you want to name it as. All your errors, all your output will be saved in this file. And then E is for error, same thing. The mail type is what if there is when the job starts, you'll get an email. If there's an error, you'll get an email. If there is no error, and if it runs throughout and it finishes the job, you'll get an email. So all means practically you'll get all the emails from the scheduler, job scheduler. The mail is your email where you want these mails to be sent. The nodes, how many nodes you want to give. Typically one node is suffice. And then how many tasks per node, usually 16 is ideal, and how much time. So this is key. I gave 48 hours and I was still not able to finish all of this in 48 hours, mainly because the same files are ridiculously big, insane. I would say about 9 to 11 GB. And for some reason, Carbonate is slow. So maybe increasing this to 2 may speed up the job. And then memory is the virtual memory you want to assign for the job. Quickly finish going over the script, assign the directory, just download the data you want for the study. So here's the link to the web page that we're using for the data that we will be analyzing. Here's a quick script that I mentioned that we will do for downloading the data to your directory. And again, these are paired end data. So when I mention slash dash dash split dash files, it will download both forward and reverse and reads. So we'll have 2 different files for one study and then 2 different for this other study, right? So treated and normal. And then we will do the fast QC to understand the quality of this read. And so after doing that analysis, you should get something like this output, the HTML file format, and you'll have to download that file to your local computer to visualize it. And so this is kind of what it shows, the Phred score, how many reads there are, the length, the GC content, etc. Yeah, so this quality score, base pair count, GC content, read length, duplication, adaptive control. And we went over all of this stuff in the first workshop. So definitely consider watching that video if you want to learn about the details of FastQC. Next, we did the adapter trimming. So we were using the trim galore trimming tool to trim the reads, to trim the adapters from the reads to get a more refined FastQ file. And again, we did the FastQC after cleaning the file. So the simple flag or tag of -- FastQC in this tool automatically runs the FastQC for us. Next, for alignment purposes, we first have to download the genome itself. And so in our case, we're using zebrafish or Daniel area. So we're first downloading the data, which is done using the wget hyperlink form command. The data can be accessed from this link here. So we first downloaded the primary assembly, and then we're unzipping it practically and first creating the indexing. So like I mentioned about the bureaus transformation and FM index, that's what we're doing right now to index the genome so it's faster or quicker to align our multiple million reads to the genome, right? So here's first we're building the indexes. We're giving the path to the FASTA file, right, which we just downloaded from Ensemble, and we're giving path to the indexes where we want the indexes to be saved. Key thing to notice here is that after we want to save our indexes in the folder assembly one, and second is we want to give the prefix to all the indexes genome underscore 105. 105 is the version number, and this is something you can keep track of when you're writing your manuscript. You can mention these details that I used a specific version number for my analysis. To perform the alignment, so once you get the index, and this usually will take about 2 hours to run. So you will get, I think, 6 to 8 different index files, and those index files are just sections of the entire genome. Right? So after you have those index files, we will do the actual alignment now. The -X flag is for the indexes that we just made, right? And 1 is for the forward end, 2 is for the reverse end. And then S is for the same file output that we are getting from after the alignment. And then this is the optional -- summary file that you can give to save the alignment score. It gives you the alignment report about cigar strings, unmapped reads, how what percent of the mapped reads, and the total alignment score. So I did the same thing for both the treated and the control sample. After aligning from my output, actually I can show you that too. So after doing the alignment, we convert the same files into the binary index format. And like I mentioned, the same files are sequence alignment map files, and BAM files are the binary index formats. These are easier to do the downstream programming, computational analysis, right? So once we have the BAM files using this command, we will sort them, because sorted BAM files are again preferred by the peak callers. It's easier for them to do the for finding their analysis. So they do a Kamer method to go through each set of base pairs or range of indexes to look for the binding regions. And it's better to have an indexed BAM files to do that, to fasten their process, right? Otherwise, they will take longer. So yeah, we sorted the BAM files. Here's just a quick information of what I try to do to compress the file and make it available for you. So if this is something that you're trying at home and don't want to do any of this because it takes some time, the job can run for days given the computation power. And if you want to directly jump to this step and start doing the peak calling and everything from there, just send me an email and I'll share this file with you. And you can extract the file using this command here and start doing the peak calling analysis from there. So yeah, once you have the BAM files, then you'll start doing the peak calling. So peak calling requires, so the peak call that we are using is max2. It uses 2 things. Well, it has other dependencies too, but primarily it uses Python 8, 8.8, and it also requires you to load this specific version of max2. And this is very specific for the people who are using Carbonate only. If you're using your local computer, you probably don't even have to, do this module load thing. You can just download max2 peak caller on your computer, and it will automatically, I believe, will download all the dependencies for the tool for your use. So once you have the tool installed, I would recommend you to make the directory max2 peak where you want to save all your output. And here's the command line that we'll use for peak calling, which is max2 call peak. T is for the treated sample, C is for the control sample, and F is the binary file format, BAM, and what's the name or the prefix you want to give to the file, to the output file. And in our case, I'm just giving treated control. There are multiple models in Max2. In our case, I'm giving no model. There's broad and narrow peaks, two options for Max2. So what it means is that it's going to look for the proteins in broad regions when you give broad flag. When you give narrow flag, it says that I'm only looking for the regions where proteins are binding is very small, right? And then I'm just giving the output directory where the max2 or the output files are saved. So quickly looking at some of these files. So now, okay, so I have ran a few of these files already. So to look at the alignment file, maybe one, not sure. Yes. So this is, I believe, the treated sample that I worked on. And this is the output. Remember the dash dash summary dash file during the alignment that we gave. This is something that will be saved. And so in this alignment process or job, we get the alignment of 92%, which is really good. This also gives us detail of how reads which align more than one time, exactly one time, 0 times, etc. Right? So you can look into the details here. Interested. Now, once we get the alignment, what we get is the same file. So let's see, R1, 9 is the treated sample. We get this same file. So same files, few of them have no sequence read. But let's look at the tail. Here's we're seeing, right, what we talked about. The sequence information, the NAIM values, and where's the sequence information? So yeah, you can look into this file and learn about all the different features. Once you have the SAM file, we convert it into BAM files, and you can see up here that I've converted these SAM files into BAM files. After converting these files to BAM file, I'm doing the peak calling, right? So for peak calling, I am saving my results in max peak, right? So let's clear this. Let's see what we have in this folder. So we have 3 things. So we have broad peaks, gapped peaks, and the XLXS file. So what we are interested in is the broad peak. The broad peaks are the one which we peak called. So what does this file look like? Treated control broad peak, right? This is exactly what we're interested in. So we have the chromosome information here. We have start position, end position, the peaks that we used. We have some sequence information here, and then we have the fold change, p-value, and FDR, like we talked about in the presentation. So what we will do next is that we will use these coordinates to get the gene information from the annotation file and we'll use that gene to do the functional enrichment, or we will use these coordinates to convert them into bed graph files. And once we get the bed graph files, we will upload that to UCSC browser to analyze expression of treated versus control samples. Okay, so that's the overall gist of the presentation. If you have any questions, please feel free to send me an email, and I'll try to get back to you as soon as I can. Thank you for watching.